Good evening, everybody. I'm Julie Passarelli. I'm the Exhibits and Collections Curator here, and it's my pleasure and honor to introduce tonight's speaker, Dr. Bengt Allen. But before I do that, I would like to introduce and recognize one of our former directors that's here tonight, Dr. Suzanne Lawrence Miller. It's kind of dark down here, but Suzanne's here. With her husband, Dr. Alan Miller, and the two of them and Bengt and I, we actually do collaborative research together. So we're really happy, and thank you guys for coming. Dr. Allen is an associate professor in the Department of Biological Sciences at California State University, Long Beach. He earned his bachelor's degree from Pomona College, his master's degree from San Diego State University, and his PhD from Stony Brook University. He is a marine community ecologist who uses observational studies and experiments to investigate links between local environmental conditions and individual organism physiology, behavior, survival, and growth. He has partnered with Cabrillo Marine Aquarium on multiple field research projects and has mentored our students in our Young Scientists program. He's a good friend of our aquarium. His work has been funded by the National Science Foundation, NOAA, and California Sea Grant. The title of tonight's talk is Ecological Consequences of Thermal Stress on Wave-Swept Shores, Insights from a Model Ecosystem. Please help me welcome Dr. Bengt Allen.
Thanks, Julie. Okay, so as Julie said, I'm a marine community ecologist. I work primarily these days on rocky shore systems, and that's what I'm going to tell you guys about tonight. I should make clear that most of what I'm going to talk about is work that's been done in my lab at Cal State Long Beach for about the last 5 to 6 years. But it's not just me that's doing this. This has been a really work of collaboration, in particular with Dr. Mark Denny, who is at Stanford University up at their marine station, Hopkins Marine Station in Pacific Grove, and Dr. Luke Miller, who is actually, some of you may know, is the son of Suzanne Lawrence Miller and Alan Miller, who were just introduced. Luke was our postdoc for 3.5 years or so on this project, and for the last couple of years has been working as an assistant professor at San Jose State University. And of course, there's been a veritable army of undergraduate and graduate students involved with this work as well. So the general context for my work is something that I suspect we're all very familiar with. It's this idea that the Earth's climate is changing, and it's changing rapidly in some fairly predictable ways. In particular, what you're looking at here is a graph. This is from the NOAA website. But what you see here is the black line is showing CO2 concentrations in the atmosphere over the last 130 years or so. And you can see they're going up, and they're going up more and more quickly. The blue and red here are the annual average global temperature measured against this horizontal line, which is the 20th century average. So what you're seeing then is what's called an anomaly. It's the deviation of any given year from the 20th century average. And so what you can see is that over the first 50 or so years in the last century, most every year was cooler than the 20th century average. In the last 50 years or so, that has flipped. And you can see, in particular, the last decade or so has most of the hottest years on record. And I think for the last 3 years, every single year has been the hottest year on record. So things are getting warmer, and most scientists, I think, agree that it is probably tied to some, in some fairly mechanistic ways, to the increasing atmospheric concentrations of CO2. Basically, CO2 is a very important greenhouse gas, and it helps keep the Earth warmer than it otherwise would be. What I think is less commonly known, although I think it's been in the news a lot more recently, is that in addition to increasing average temperatures, we're also starting to see increases in temperature variation. In other words, what you're seeing here is, and you can see it bounces around quite a bit, but over the last decade or so, there appears to be some increasing trend for the frequency of extreme weather events. And I will just tell you that all of the models that are projecting out what might happen over the next 100 years or so all show significant increases in temperature variation as well as changes in the average. And that's sort of interesting because we don't actually know very much about what to expect with changing variability in a system. All of our statistics, most of our models are all designed about what's going to happen as the average changes, or let's compare the average of this group to the average of some other group. We don't really deal or think about variances very much. And so this was really sort of the jumping off point for a lot of what I'm going to talk about today, which is, well, what happens, even if you ignore changes in the average, what happens to biological systems when you start changing the variability? And we're already seeing biological effects of global change. We've seen changes or shifts in the geographic ranges of organisms where we find things around the globe, changes in phenology, which is the timing of ecological events, changes in species interactions, and even mass mortality events due, again, in part to some of these extreme conditions. And there are some, we do know, as I said, a little bit about what to expect with increasing variation. And one of the things that's I think fairly interesting and not something I think most biologists even are very aware of is something that has to do with the shape of the performance curve or the relationship between organismal performance and temperature. So thermal performance of ectotherms, which are animals or organisms that don't generate much of their own body heat, their body temperature is really determined by their local environmental conditions. And so for many of them, as the temperature increases, their performance increases up to some point at an intermediate temperature, and if it gets much hotter than that, performance decreases again. And so it looks something like this. So here we have performance, and it doesn't really matter what it is. This shape of this general curve is true for lots of different things you might measure. If we look at body temperature here, you can see that at very cold temperatures, performance is pretty low, and that's because most biological things, chemical reactions and so forth, don't really function very well at cold temperatures. As temperatures warm up, things do better, up to some point at some optimum temperature where you get maximal performance, and then at temperatures above that, you see this very rapid drop-off. So ignore the math. I know it's Friday night, and that's probably not why you're here, but this is a pretty simple concept. And so what I want you to think about is if you have a given body temperature, let's say it's the optimum temperature, right? If we think about this as a function, here's the function, performance is a function of temperature. If we put the input into that function as some given temperature, we get an output which is the level of performance. Is everybody with me? Great, so that's not so bad, right? What's interesting is if you add variation to the input, even if the average is still the optimum temperature, what happens is because of this nonlinear performance curve, is that when you are at any temperature other than the optimum, so here, for example, the performance value you get out is much lower. Same here, it's lower. And so when you look at the average outcome or average performance level, due to this variation in input in temperature, the average outcome is now lower than it would have been if there was no variation, even if the average temperature is the same. So that's just a consequence of this nonlinearity. It's something called Jensen's inequality. You don't need to know that. But the main point is that even all else being equal, if the average stays the same but the variation in temperature increases, we might expect to see, in this case, reductions in performance, which is interesting. The other thing is that increases in temperature variability are also going to presumably lead to higher or more extreme values. And if you think about it for about 2 seconds, you realize it's not the average day that's going to stress you or kill you. It's the hot day, the really hot day, or the really cold day. And so if you think about, here's an example looking at temperature over time. This would be for an organism, for example, in the intertidal zone. So here we have the tide height, and you can see it goes high tide and low tide every day. When the low tide happens in the middle of the day, you can see the organisms on the shore are now exposed because the water level is low. They're exposed to sunlight and wind, and they tend to heat up. And you can see then that as you increase the variability in temperature, you increase the likelihood of exceeding some threshold, stressful or even mortality-inducing threshold. So that's another way in which increasing variability, even if you leave the average pretty similar, might be problematic for organisms. And in particular, I mentioned that example was sort of a rocky shore. That's a really great system for asking questions about the consequences of this kind of change. Because during aerial exposure at low tide, the body temperature of many of these organisms, which are mostly ectotherms, so again, they're really determined by the local environment, they can increase by 15 or 20 degrees Celsius, which is really a lot. If you think about it, when they're covered at water and high tide, they're in this nice cool environment. The water goes down, the sun comes out, and they can heat up a lot. And they have to deal with that every day, and twice a day sometimes. And we already know from other work that people have done that many of these species are already at or near their tolerance limits. They can't really get much hotter than they currently do. And that they may not be able to acclimate or sort of adjust physiologically much beyond that. They appear to be sort of near their limit already. So we would expect to see, even with relatively small changes in environmental conditions, some sort of biological effects. And just to show you, this is a thermal image here of a black abalone. So here's a black abalone. This is generally an organism that's found pretty low on the shore. Often you can see this one's halfway in a crevice. But even so, if you look at the temperature of this thing, this is almost 27 degrees. It's almost 10 degrees warmer than water temperature. And this is not a high intertidal, really exposed animal. And you can see that it still can get reasonably warm, and this was just not a very hot day either. It was sort of a moderate 75 degree day. So this is a system that is probably pretty perfect for asking some of these kinds of questions. And so what I'm going to talk about a little bit today is sort of three different stories that I think are hopefully, you'll see how they're relatively connected. And there are different kinds of studies on different kinds of organisms, but again, all in this big context of what happens when the environment, and in particular temperature, gets more variable. Our basic question, and this leads out of that thermal performance curve we just talked about, is does increased temperature variation lead to reduced average performance, as we might expect it to? And so when I say we, in this case, this is Mark Denny and Luke Miller and I, in particular, working on this. And so we investigated these effects of temperature variation on physiology and growth of a suite of conspecific limpets. That basically means a bunch of limpets are all in the same species, or sorry, in the same genus, but they're different species. And so they're relatively closely related evolutionarily, and they often sort of do basically the same thing. Limpet, for those of you that aren't familiar, is basically a small flat snail. And they live on the shore, and they scrape algae off the rocks. And we looked at these 4 species, and this work was done up at Hopkins Marine Station in Pacific Grove. So these are 4 species in the genus Laudia, and you can see that they are divided by where they live on the shore. So someday I'll get that right. So here we have Laudia limacula and Laudia pelta are found sort of low on the shore. Mean low water is just a measurement of tide height. That's the average of the low tides for some stretch of time. And so we generally call that 0, and then we measure everything relative to that in terms of shore height. Higher on the shore, we have Laudia scabra and Laudia ostrodigitalis. And even these two, although they live in the same basic zone in the high shore, they actually partition the habitat a little bit. So ostrodigitalis tends to be found on vertical surfaces, whereas scabra is found on more horizontal surfaces. The horizontal surfaces get the hottest. They're the most exposed to direct sunlight. So as you go higher on the shore, you expect these things to experience hotter temperatures, and then scabra in general would be the warmest. And so we had sort of a couple basic hypotheses when we started this work. The first was that limpet physiological responses to temperature would be species specific. In other words, they would vary between the species, and they'd be correlated with where we find them in the field. Those species, the limpets found highest on the shore, we thought would be most tolerant to high temperatures. We also thought that because of this Jensen's inequality idea that maybe limpet growth rates would be negatively related to body temperature variability. And so, well, we went and set out to try to investigate that. And so the first thing we did was just super simple kind of baseline measurements of respiration, which is sort of an estimate of metabolism. We just wanted to see how their oxygen use, energy use changed as a function of temperature. And so we did that by measuring the rate at which they used oxygen in in a closed system here. So we put these limpets in these little wells, we controlled the temperature, and then we just measured how much they were using oxygen over a certain amount of time. And what you can see as a function of temperature. So we did that both in water, which is the open symbols here, and then in air, which are the closed symbols here. And you can see we've got the 4 colors for the different species. And the basic pattern is, first of all, I'll just point out that this curve is very similar to what we had just talked about, right? Very low levels of performance, in this case, respiration, low levels at low temperatures, increasing with temperature up to some optimum, and then the fall off as they get stressed and as the temperatures get too high. You'll notice that the curve, the peaks where the fall off starts to happen are pushed out to higher temperatures for the red and blue limpets. Those are the Scabra and Ostradigitalis, which are the high shore species. You'll notice also that the slope of these lines are lower for the high shore species, which means they can control their respiration rate, they can limit their energy waste as the temperature increases more efficiently than can the low shore species, which presumably don't actually have to deal with high temperatures all that often. We also looked at the what we call the heat coma temperature. So this is basically just a measure of thermal tolerance, where we basically, you measure the, and we're calling it survival, but we don't actually know that they died. But these were limpets that at the end of our temperature experiments were not responsive and weren't able to stick on the wall of the chamber anymore. So they were not happy at any rate. And so we asked, what temperature do we see 50% of our sample limpets exhibit this heat coma behavior? And what you can see is, again, the high shore species, the temperature at which that happens is several degrees warmer than for the lower shore species. So with respect to our first hypothesis, I think we found pretty good evidence that, yes, the species differ physiologically in terms of their response to temperature, and it's in pretty much ways you would expect. Evaluating our second hypothesis, though, about the growth rates was a little bit trickier. We had to figure out how can we know that there's differences in the variation in temperature that these things are experiencing over some long enough period of time that we can actually relate it to growth. And so what we ended up doing was manipulating where the limpets were out in the shore by putting out these aluminum plates that kind of almost look like a little hockey puck with some grip tape on top, the anti-skid tape you would see like on a boat or something. So it's kind of nubbly and it sort of looks very similar to the rock surface. We put fences around it so the limpets either couldn't crawl on, or you can see we put some limpets on it, they couldn't crawl off. And then inside we milled a little pocket, and I mean Luke, basically made hundreds of these things. And we put an I-button temperature data logger. So this is a thing, it's about the size of a stack of maybe 3 or 4 dimes, but it actually records temperature. And you can program it to do it as often as you want. We had it doing it every 12 minutes. We would get a temperature reading for each plate. Here is what it looks like when we coated it in electronics wax to keep the seawater from wrecking it. So we would put one of these in each of the plates, and then that would give us we have basically a 7-month temperature record of that plate taken every 12 minutes. And then we can track limpet growth and loss and things like that and relate it to the temperature record. These are some of the students from Stanford and Long Beach that helped set this up. And so we put them out for 7 months, and then we looked at growth. And it turns out that food, the algae that grows, you can see these plates are kind of green looking. That's the microalgae that's growing on there that the limpets will scrape off. And what you can see, what it turned out is that the amount of food, not surprisingly, actually mattered in terms of the growth rate. So we actually separated out into low, medium, and high amounts of algae. And that's what you're seeing here. So what you have, this is limpet growth rate in micrograms per day. So basically, how much tissue are they adding as a function of the average daily maximum temperature here of the plate. So this is over this is on a monthly basis. We just asked what was the average daily maximum temperature on that plate for a given month, and how much did that limpets on that plate grow. And we split it up by species. So here are our 4 species. And you can see that in general, for Pelta, there was really no pattern at all. The temperature didn't really affect growth. They just didn't grow for the most part. But Ostradigitalis and Scabra, yeah, maybe there's a slight hint of some increase in growth. But that as we increase the amount of food, the ability of these limpets to take advantage of that was really shocking to us. In particular, some of these species, the high intertidal species here, really grew great. What was our hypothesis? Increasing temperature variation. And again, the thing is, this is all relative to baseline, it's water temperature. So every day this gets set to cold water temperature. So it's really a question of average daily maximum is how hot did it get during the low tide? And then it goes back down to water. So the most variable temperature plates are up here. And what we got was crazy high growth, which is pretty much the exact opposite of what we expected, right? We thought growth would go down. This bothered us for a while, because this was not really what we expected. It's not at all what we thought. It turns out, we think, is what's happening. There was two things. The first is it's never actually all that hot at Hopkins. This is Central California. This is summer. Turns out it's actually not the best time of year to be necessarily doing this kind of experiment. If we look at, these are the just a density function, a probability of seeing a given temperature on two different plates. One that in the blue here is the low variation site. One was a hotter site in red. But you can see, even so, the hottest temperatures are just very, very infrequent, right? If you sum up the entire 7 months period of time, there's just not that many hours when they're really very hot. So we probably weren't actually catching much in the way of stress to measure their response. The other thing is, if you think back to this curve here, the performance curve, this whole stretch right here, as you increase temperature, you actually see an increase in performance, right? We sort of were focusing on this part, but in fact, I think, and if you look at the actual numbers on the temperature, the temperatures on the plates, I think we're down in this region. Water temperature here is cold. They probably do better by warming up a moderate amount. And in fact, what we think is going on, and I have to admit, I didn't ever have lizards or snakes growing up. We had like dogs and rabbits and birds and everything else. Does anybody here have a lizard or a snake? What happens when you feed them? They tend to go thermoregulate and maintain a high body temperature for a while. And that maximizes their digestive efficiency. And so what we think is maybe happening is these things are feeding at high tide when they're covered by water, so they have a belly full of algae. And then when the tide drops, if they can warm up a little bit, we think that their efficiency and growth actually increases as well. So yeah, that wasn't what we were really interested in. We really wanted to know about the hot stuff. So we'll come back to that in a second. So basically, though, we did conclude that species found at more thermally stressful environments did like we expected, were more tolerant of these high temperatures, that stressful conditions are actually not that common currently at Hopkins at any rate, and that moderate warming might actually be beneficial, not actually bad like we were thinking it would be. And the other thing is, as I mentioned, that food obviously made a difference. If they had more food, they were able to respond more strongly. Again, not really shocking, but it's always good to note. So the thing is, well, our whole context, right, is that the Earth's climate is changing and it's warming and it's going to keep doing that for the foreseeable future. And so, our current conditions might not actually be the best set of conditions to test this, our basic idea. So what we decided is that we needed to figure out how to manipulate temperature variation in a way that would allow us to see some of these more extreme situations, so that we could really get at this idea of what happens when you start getting into the other side of the curve. And so what we did is we did that. We manipulated the temperature of another set of plates here. And so what we did is, this is the plates I described to you previously, what we're calling the natural temperature variation, where it's basically this aluminum plate. We had a little thin film of concrete underneath it just to make a nice flat surface that we could bolt it to. So bolted directly to the rock, that sort of, the plate temperature is rock temperature because the aluminum is a really good thermal conductor. And then limpets that are sitting on that plate, because they have this really big foot that they use to hold on, they're basically plate temperature. We then took some closed cell foam insulation and we stuck it under some of the plates, which essentially isolated the plate away from the rock, and so it tended to heat up more during low tide in the sun. And then we drilled out a chunk of rock and put in an aluminum rod that we cemented into the bottom there, and then bolted the plate to that. And these plates are actually in thermal equilibrium with the rock about a foot beneath the surface. In other words, those never warm up very much. So we basically created a much broader range of plate variation, and then asked how that affected the limpets. So here is a CSULB student drilling giant holes in the rock up at Hopkins. And you can see that, yeah, they made it look like Swiss cheese, basically. And then we bolted these plates to it and asked what happened. You've seen this figure before, but this was actually from some of our plates. And what you can see is that we were able to adjust the temperature variability on these plates pretty significantly. In other words, the manipulation actually works. And so then we, again, put limpets on the plates and then monitored their performance over time and asked how they did. And in this case, we were able to generate much higher values here. We got into the hotter, more stressful side of the curve. And what we saw was actually kind of interesting. If we looked at Laudia scabra here, again, this is the one that experiences the hottest temperatures. They mostly didn't really, this is looking at loss rates, so how many limpets disappeared from our plates any given month. And scabra mostly didn't. They just kind of stuck around. They didn't seem too bothered by temperature. On the other hand, Ostra digitalis always had higher rates of loss than scabra, and they increased with increasing temperature. Now, we don't know, we're not calling this mortality because we don't know that these limpets actually died, although it's possible some did. But for whatever reason, they were motivated enough to get off the plate if it got too hot. So we do know that at the very least, they were bothered by the high temperatures to a greater degree than was scabra. More interestingly, I think, is we look at growth rates here. Here we see, again, this increase in growth initially with a moderate warming, like we saw before. And then with scabra, there was really no, this is just no data. None of the plates that we got limpets off of got all that hot for this species. But basically, they didn't seem all that bothered by high temperatures. On the other hand, Ostra digitalis, you can see there's an initial increase in growth rate with moderate warming, and then basically as it got hotter, they grew less and less and less. And again, Ostra digitalis is exposed to less stressful temperatures in general than scabra, and so that pattern is at least consistent with that idea of where we would find them on the shore. What we think is happening here, and these are data that were collected by Yunwei Dong, who was a postdoc of George Somro, who's another faculty up at Hopkins. He's a physiologist. And what they were looking at is relative expression levels of HSP70. That stands for heat shock protein 70. It's just it's an enzyme that many organisms produce in response to stress, in particular thermal stress. And it helps basically maintain physiological function at temperatures that otherwise would be problematic. So it's a marker for these things might be stressed. And what we see is if we look at the pattern of expression, and this is a log scale here, so these differences are actually pretty extreme. What we see is that Laudia scabra, the organism that's mostly exposed to very high temperatures pretty regularly, they have high levels of this protein expressed all the time. That's what we call a constitutive defense. It's always on. In other words, they're never going to get surprised by a hot day because they're always ready for it. They're always prepared. Whereas Ostra digitalis has a very different thermal defense strategy. They basically don't make much of this protein, and as long as they're not exposed to very hot temperatures, they're fine. They don't need it. On the other hand, if they do get exposed to higher temperatures, all of a sudden they'll switch that production on, and they'll generate all of a sudden much more of that protein, so that now they are protected. And the advantage of what's this so-called inducible strategy is that you're not wasting energy here making a protein, a defense that you don't actually need. And so we think that this pattern of inducibility at the high temperatures is probably explaining this reduction in growth at the higher temperatures. In other words, that here the individuals are experiencing hotter temperatures, and so they've turned on this machinery to make a defense, but it has a cost, right, in terms of their growth. That's a nice story, and it might even be true. The problem is, we also took other data, and in particular, we used this. It's called a PAM fluorometer. You basically shine a light out of this fiber optic here, and then it reads the fluorescence coming that's emitted off the plate, and we can basically calculate or use a calculation to convert this measurement of light coming off the plate into a measure of biomass of chlorophyll. That's the pigment that these microalgae use to photosynthesize. Basically, the higher the fluorescence value, this FO, the more algae there is on the plate. And what we found, and again, this is food for limpets, basically. And what we found is that as you increase the plate temperature variability, we see a pretty strong decline in algae. In other words, there's less food on the hot plates. So the fact that the limpets are growing less on the hot plates might be because they're spending energy dealing with the stress, or it might be that there's just less to eat. So that's a problem. We did a little sort of side project just to see what was happening there, and so we put the plates inside this chamber here, and we measured the uptake rate of carbon dioxide, which is a measure of photosynthetic capacity. And what we found is that if we expose plates during a simulated low tide in the lab to one of different, several different peak temperatures. In other words, we heated them up a little bit or a lot. We measured photosynthesis before we did it and then afterwards, and then we asked, was there any change in photosynthetic rate before versus after this exposure? And what you can see here is that, so 0 would be they were photosynthesizing at exactly the same rate before versus after the simulated low tide. And at cool temperatures, that's basically what we found, no real change. But as you got hotter, you see these more and more negative effects. In other words, they were performing much less well after exposure to high temperatures than they were before. And so this would, because photosynthesis is essentially how these algae grow, over time you would expect to see that if this pattern holds, that you should end up with less algae on the hot plates, and that's consistent with what we saw. Yeah, high temperature variation can have negative effects on limpet persistence. In other words, how likely we are to find them month to month in a given spot, and their growth. Species-specific responses might differ depending on their thermal defense strategy. And again, local resource levels appear to be important. The problem is that this is a little, we can't really disentangle this in the field. They're correlated. So the hottest areas of the shore are also the ones with the least amount of food. And so that's kind of a problem for us because we can't really definitively say, is it food or is it temperature or is it both, and if so, how much of each? So we're going to leave off with that study for the moment and switch to a different system. These are actually work done here down at Abalone Cove, just here on the peninsula, and using mussels instead of limpets. And the reason is because this turns out to be a better system to answer our next question, which is basically jumping off of where we left, which is the activation of a thermal defense, for example, this heat shock response, is that energetically expensive in a way that imposes what we call a life history trade-off? In other words, energy you spend doing one thing is energy you can no longer spend to do something else. So you can invest in a thermal defense that increases your chance of survival, but it has a growth penalty, for example. And therefore resources or amount of food, energy that you have available then is going to be important. And so what we wanted to know or ask this question is there this trade-off between thermal stress defenses and growth? And we decided that we couldn't really figure out how to do that or address that well with the limpet system. But what we could do is in the lab, we could manipulate food and temperature stress to mussels in a much more controlled way. And so that's actually what we did. We switched organisms to the California mussel, Middleus californianus, and they're actually a pretty interesting animal in their own right. They're the dominant space occupier in the mid-intertidal zone, so they outcompete everything else given the opportunity. They provide habitat for a lot of other organisms, and also strong benthic pelagic coupling. In other words, they feed on plankton that's floating in the water. They filter it out, and so they're taking energy that's produced in the water column and they're putting it onto the bottom, onto the benthos. They're also important in that lots of things eat them, and so they're important for moving energy through the food system. And they're also just easy to manipulate and move around, and we already know from previous work that they're sensitive to high temperatures. So it's a pretty good system to investigate this question. And we also know, this is just data from actually Abalone Cove, where if we look at mussels growing across a wave exposure gradient here, as a function of average daily temperature, you can see the growth rate goes down, which again is consistent with this idea of thermal stress imposes a physiological cost. But again, the problem is this is a wave exposure gradient, so the hottest sites tend to get splashed the least, and so again, less opportunity for feeding, perhaps. So we really can't disentangle those two questions in the field very easily. But we can in the lab. And so we set out to test these two hypotheses. One was that relative to unstressed individuals, muscles exposed to chronic temperature stress, in other words, repeated temperature stress over time, would exhibit a trade-off between thermal tolerance or survival and then growth or condition. Condition meaning just sort of size-specific tissue mass, stored energy reserve, something like that. So generally, if you're in better condition, you have more capacity for growth is the thinking. We also thought that increased food in general would lead to higher performance, higher investment in both things. In other words, if you have more to eat, you can be both better defended and grow more. And so we investigated this in the lab, as I said. We collected animals from the shore. We brought them back to Long Beach to the marine lab there, and we held them in this flow-through recirculating sea table for 8 weeks. And we had 4 different treatment groups. We had what we called a high stress and a low stress group, and then a high food and a low food group. And so the food one's pretty much just like it sounds. The stress group was basically that every day all the mussels got exposed to a simulated low tide. They were taken out of the water. Half of them were just left in the cool bench. They never got very warm. The other half were put under heat lamps, and their temperatures were raised up to 32 degrees. And again, that happened every day. And so over 8 weeks, then they either dealt with that or they didn't, I guess. And at the end, we measured shell and tissue growth and asked what happened. We also, at the end of 8 weeks, then took those surviving mussels and we exposed them to an even more extreme thermal challenge. So we used these heat lamps here and we raised their temperatures to 36, 39, or 42 degrees, which is really pushing them right up to the edge of their thermal tolerance. And we asked how did they survive, and did the fact that they were, or did their prior thermal history or prior food ration have any effect on their survival. And so here's what we found. Basically, we're looking at dry tissue mass, so this is our measure of condition. This is size specific. So for a given size muscle, how much energy reserves does it have? And this is a function of their chronic stress level, low or high, or their food level, high or low. I'll have to figure out why that should flip that around, but it doesn't matter. The main point is, if you look at both groups, whatever the food level, high food or low food, the stressed individuals in the hatched bars here always had lower condition. Right? In other words, they had lost tissue mass over the 8 weeks in a way that the others had not, suggesting that there's some sort of energetic cost to dealing with that chronic stress. On the other hand, if we look at the survival here, so proportion surviving after exposure to 36, 39, or 42 degrees, we see two things. One is fairly intuitive, I think, which is that the hotter the temperature, the less overall survival, the lower the overall survival. What's more interesting, though, is this pattern, which is that basically now, again, if we look at chronic stress, low and high, and then the food levels, that in this case, survival is higher for the organisms that had been stressed before, right? So they had lower condition, but better survival. So there's a trade-off. And so, so I think in terms of our first question, is there a trade-off? Yes, there is. They have less tissue or less growth potential if they have to deal with chronic stress, but they're more able to tolerate a future stress. And the second question about what's the effect of food availability, that was pretty straightforward. In general, on average, you had a better tissue mass if you were in the high food group than the low food group, and you had better survival if you were in the high food group versus the low food group. So in general, having more food is good. Okay. So in general, then, our data here, I think, are much cleaner and much more interpretable than the field data. And they're definitely consistent with this idea that tolerating sublethal stress has a cost, but that it has some benefit or some potential payoff as well. And that individuals with limited access to resources, then, are basically really sad because they can't do anything well. And they're at much greater risk of stress or mortality. And the implications of that, then, are that, again, coming back to this bigger context, if we increase the frequency and intensity of these thermal stress events in the future, that probably is going to have some fairly significant effects on marine intertidal populations. And I'll just remind you that, many of these invertebrates, the amount of reproduction they can do, the number of offspring they can produce, is entirely dependent on size. Size also influences your resistance to vulnerability to predation, for example. And so, if we have smaller populations of smaller individuals that are producing fewer offspring, that over time will have consequences for community structure in these systems. And then, of course, the magnitude of these effects appear to be moderated to some degree by food level, by resource availability. And so, I will just say that at the time we published this paper a couple years ago, nobody was really pointing this out. Like, there were lots of models about what's the effect if we know something about the temperature biology of organisms and we know something about how the temperature is going to change, we can make predictions. It's like, and what we want people to take away from this, at least in part, is yes, you can, but you need to know something about the local resource level also. Because if you don't, then your predictions might not be worth very much. This work is actually still ongoing at this point. We have taken the mussels from that lab experiment, and we actually are working with Lars Tomanek, who's faculty up at Cal Poly San Luis Obispo, and he does what we call environmental proteomics. So it's basically, it's a biotechnology approach that allows you to analyze protein levels in a really complex way. So what he's done for us, I'll stop that someday, is we basically, he grinds up the tissue of the mussels, extracts the proteins, and then runs them out in this two-dimensional gel. So each dot that you see here is a different protein, and those numbers are ones that they've identified as being discrete spots. You can then pull the spot out of the gel, run it through a super expensive mass spectrophotometer, which is kind of magic that I don't do, and you can figure out what protein it is. So he identified 330 spots, or found 330 discrete spots. He was able to identify 84 of them that actually showed changes, and this is what a subset of this data looks like. And you don't really need to worry too much about the specifics here. You can see these are our 4 treatment groups: high food, low stress; high food, high stress; low food, low stress. Each column is actually an individual mussel, and the map goes from blue to orange, blue being not very much of this protein, orange being a whole lot of it. And here are some of the protein identifications. Honestly, I don't know what most of that means. But I know what the functions of some of these are. So some of these are growth related, some of these are chaperones, or like heat shock proteins, for example. That's the HSP22 and 24.1. So these are stress related proteins. And you can see, just looking here, there are patterns, right? There are some of the groups have more of certain kinds of proteins, some have less. And so what we're working with him is to try to get a mechanistic understanding of exactly what's going on with these mussels that are dealing with different levels of stress in food. And sort of our first, as I said, we're still doing this, but this is a graph that shows some of the patterns or summarizes them in a more maybe viewable way. The way to read a graph like this, this is a single mussel, each dot, and it is the whole complement of all 84 proteins that are sort of summarized in the location of that dot on this two-dimensional space. And so basically, dots that are close together have very similar protein patterns. Dots that are far apart have very different ones. And you can see pretty easily, I think, that these 4 groups cluster out quite separately. So in other words, the protein expression patterns of these different of mussels in these different groups is quite different. And so, as I said, we're sort of sorting through mechanistically what exactly is happening there. But I think hopefully it'll be pretty interesting when we do. All right, so the last section I'm going to talk about here is sort of where we're going next. So that's all stuff that's mostly been done. It's either been published or we're writing it up now. And so the question is, where do we go now? And mostly what we've been doing up to this point is really focusing on sort of individual species and looking at, or even individuals, and how they are responding. But the reality is, that doesn't necessarily tell us all that much, because species are not usually responding to just a single factor in their world. They're interacting with other species as well. And so the next step really is to start to broaden out our questioning about how we can sort of anchor these results in a broader natural system or natural context. And so one of the things that we've become fairly interested in recently is this idea of compounded perturbations. So perturbation is just like a disturbance or sort of a stress event. And so what we know, and again, this is from lots of work people have done in lots of different systems, is that disturbances that are sort of in the typical range that an organism might deal with in its lifespan, they tend to not necessarily have huge effects over the long term, right? Because presumably these organisms have adapted to deal with stresses of that general magnitude. It's when the magnitude changes, right, which is sort of our how we first started asking about what happens when the variance increases, that you start to see issues. The other way that you often see responses is when you get more than one type of stress happening at the same time. So in other words, you might be okay responding to one stress, but if you get knocked by a second one at the same time, you might not be able to deal with it very well. And so that's sort of an interesting question, I think, for us, is this idea of how do things deal, what are the effects of multiple stressors? And in particular, the one that we're focusing on right now is what we call ocean acidification. So ocean acidification has sometimes been referred to as the other carbon problem, right? We talked about the increasing atmospheric concentrations of CO2, of carbon dioxide, are leading to warming temperatures. Increasing atmospheric concentrations of CO2 are also leading to increasing dissolved levels of what's called PCO2, or the partial pressure of CO2 in seawater, surface seawater. That is potentially problematic because in seawater, carbon dioxide enters what's called the carbonate buffer system. And it basically, there's a series of chemical reactions with water that form carbonic acid, which then becomes bicarbonate, and bicarbonate and carbonic acid, as that process happens, release, and eventually to carbonate, release hydrogen ions, H+. And if you remember your basic chemistry, hydrogen ions, or the concentration of hydrogen ions in solution, is a measure of acidity, or the pH, right? And so what's happening is that pH is decreasing, the oceans are becoming more acidic over time because of this input in carbon. And what happens is if you have a calcium carbonate shell, as many marine organisms do, including limpets and mussels, and abalone and all sorts of other things, calcium carbonate tends to dissolve much more easily in low pH. So in normal pH water, we'd have normal shells. In acidic, low pH water, we tend to have deformed shells. And so that's a problem. And we think that is probably an energetic issue, that it's energetically more costly to try to make and keep a shell in the face of low pH conditions. And just to show you that this is not just a theoretical thing, this is actually measurable and it is happening. This is the CO2 data in a more granular form that we just looked at on that first slide. So this is in red, the atmospheric concentration of CO2 measured at Mauna Loa in Hawaii. And basically you can see it goes up and down within a year. That has to do with the seasons in the northern hemisphere. In the spring and summer, all the trees and grass and things that are photosynthesizing pull in a lot of CO2, so the atmospheric concentration drops. In the winter, everything is respiring and releasing CO2, and so the atmospheric concentrations go up a little bit, but they bounce around. And you can see in general, though, they're trending up in the atmosphere. And then in blue here, we have the seawater PCO2, which is the concentration of CO2 in surface waters. And then we have the pH in green. And so as atmospheric concentration of CO2 goes up, surface water concentrations go up, and pH goes down. So as a sort of follow-on from the work we've already done and our sort of direction now of where we're going is we're interested in trying to combine those two things. Like, okay, well, we know a bit about temperature variability. What's the consequence of that interacting with this additional stress of low pH? And I should say that most of the models right now predict that we're going to see changes. So current pH is typically 8.1, something like that. That's kind of normal ocean seawater pH. And the expectation, the model suggests that by the end of the century, we might be down at maybe 7.6, 7.8. And again, this is a log scale, so that's actually a fairly significant change. But that's, again, 100 years out, say. And there have been a lot of lab studies that people have done where they've manipulated CO2, they bubbled CO2 into seawater, and then they ask how that affects organismal performance. Some of them have also changed water temperature and asked how that affects things. There's basically been really one, maybe two studies at this point that have looked at the effects of temperature exposure during low tide, right, which is what we were focusing on, because that actually is going to be much more stressful for things than water, than small changes in water temperature. And none of that work's been done in the field for the obvious reason that current pH is not anywhere near what is predicted for the end of the century in most places. It turns out there is actually a place where that's not true. This is Northern California. Here is Humboldt State University up near the Oregon border. And there are a series of intertidal sites in that region, some of which get exposed to upwelling water very close to shore. So upwelling is a process where winds blow surface waters offshore, deeper waters come upwell from below to fill in, and some of those upwelled waters are very acidic because they have a lot of carbon in them. And so it was shown in 2008, somebody did a series of tracks where they showed, they monitored pH in surface waters near shore, and they found that in this region near Humboldt, there are areas of upwelling that are very close to shore because the continental shelf is pretty narrow there, and these waters were very, very low pH. This is actually data from, this is Trinidad is the Humboldt's has a marine lab about 15 minutes north of campus. This is Trinidad Pier. There's been a pH monitoring station off the end of the pier there for the last, well, since 2009. And you can see this is the average daily pH. So it's not the minimum. This is just the average for every given day over for the year from January to December over 9 years. So the black line is the average for the whole 9 years. The gray is the 95% confidence interval, in other words, sort of the bounds of variation that we would expect to see. And you can see that during the winter, it's up at about 8, which is 8.1, which is what we'd expect. And in the sort of spring, from about April to September, it can get to be really quite low. Actually, end of the century predicted low right now. So what that means is that this might be an area we could actually study the effects of pH, of low pH, on intertidal organisms in the field, which is pretty cool. So that's what we're starting to do. One of our hypotheses is that, based on lots and lots of lab studies, is that mussel shell thickness and breaking strength should be lower under high PCO2, low pH conditions. Again, because the shell dissolves more easily, they have less energy to spend to maintain it. And our assumption is that any negative effects of OA, of ocean acidification, will be made worse by the addition of temperature stress on top of that. And that one of the interesting things, though, is that upwelling, as some of you may know, is that water that comes up from down deep, in addition to being very low pH, often has a lot of nutrients. In other words, upwelling zones are where we get a lot of primary productivity, a lot of phytoplankton produced, phytoplankton being the main food source for mussels. So it is possible that there, in these upwelling zones, that even though it may be stressful, the addition of lots of extra food might give them the resources that they need to deal with that stress, and so actually maybe there aren't negative effects. And so that's one of the interesting reasons to do stuff in the field as opposed to just manipulate things in the lab and then make extrapolations. And so we're proposing at least that energetic subsidies associated with this upwelling might actually counteract any negative effects of OA and perhaps thermal stress as well. So, okay, what can we say about this first idea, about mussel shell thickness and breaking strength will be reduced under low pH conditions? Well, we actually went out to 4 sites near Humboldt, 2 just to the south in Mendocino County, Belinda Point and Devil's Gate, where near Cape Mendocino, and these areas really don't experience that upwelling to any great degree. They generally have pretty stable, pretty high, normal, what we call ambient pH conditions most of the time. And then there were 2 sites up, Baker's Beach is right near Trinidad, where we just looked at the pH data from the pier, and then Point St. George is up near Crescent City, so a little bit further north. And basically what we're looking at here is average daily pH at these sites, and you can see that at the southern sites, they're generally up near 8, and they're not that variable, and then at the northern sites near Humboldt, pH is quite a bit significantly reduced, and particularly so at Point St. George. If we look at chlorophyll, which is a proxy, we can look at the amount of chlorophyll that is in seawater, and that sort of correlates pretty nicely to the amount of phytoplankton, the amount of food for mussels. And if we look at the upwelling sites, we can see that there's actually much more chlorophyll there than there is in the ambient sites. So, so the upwelling regions pretty much look like we think they should. If we ask then, what's the effect on mussels? So yeah, what I just said. What's the effect on mussels? It's actually pretty interesting. So this is shell thickness of mussels that were collected at each of these 4 sites. And you can see that in general the shells are pretty nice and thick down south, and they're much thinner at Baker's Beach, but not actually at Point St. George. And remember, Point St. George is where they have a ton of food. And the same is true for the shell breaking force. There's a machine called an instron that you put the shell in and then you basically just mush it until it breaks, and then you measure the force it took to break it. And you can see the breaking force is relatively high for the mussels from the south, and it's somewhat less so and lowest at the Baker's Beach site, which is the upwelling but not as much food. So it appears as though that I guess if we go back to our hypotheses, that mussel shell thickness and breaking strength are in fact lower under low pH conditions. We don't actually know anything about this yet because we didn't look at temperature, so we'll get back to you on that. But that lower negative effects of OA on shell strength may be mitigated by the presence of food. And so clearly there's a lot going on here, and these are all just sort of snapshot we went out and sampled once. And what we need to do now is actually do manipulative experiments where we control things. We look at growth over time. We look at the effects of these changes in mussel performance on their interactions in the broader community. And that's actually the final bit here that I want to talk about. These effects on mussel, any negative effects on mussel growth rate, on mussel size, on mussel shell strength, are probably going to have consequences, because at least at these sites, crabs are the primary predator on mussels, and mussel size and mussel shell strength, thickness, are the 2 primary defenses mussels have. If they're big enough with a thick enough shell, the crabs can't get in. But if they're small or weak, the crabs can. And you can see here, this is just showing that this is the number of prey eaten per hour or per 6 hours. So they were offered some snails, Nucella and Ategula, and then Middleus, the mussels, and the crabs strongly prefer to eat the mussels because they get a lot more tissue for their effort. And then this is just to show that out in the field, if you cage the mussels, they survive just fine. There's a number of mussels lost per day. But at least in the low shore, if they're not in a cage, they get eaten pretty quickly. And we have a lot of evidence showing that that's actually crabs that are doing the eating. It's possible that normally up in Northern California, we would actually expect to see Pisaster ocraceus. That's the purple or oak or orange sea star that we used to see out here. Some of you probably know that there was a wasting disease a few years ago and a big die-off along much of the West Coast. And so in this case, we don't really have data from before that, so we don't know if there used to be sea stars here and now there's not, and that's why the crabs are such an important predator. But at least for the moment, we know that the crabs matter. And so the effects of OA, of acidification on the mussel shell is probably as important ecologically. So to summarize all of this, because that's kind of a lot, that's a lot of data, in general, along with these positive effects on survival, acclimation to stressful conditions has trade-offs or has consequences that might lead to reduced performance in other dimensions, for example, growth or reproductive output. Energetic subsidies, on the other hand, may, because these are energy-related trade-offs, if you have more energy available, you might not see such negative effects, and so they might be able to alleviate, or energetic subsidies might alleviate some of that. And I guess the other thing I would want you to take away is that this is really complicated, right? It's like trying to, I mean, it's like, each piece by itself seems pretty straightforward, but once you start trying to fit them all together, it gets tricky. And I think I would argue that, just working in the lab or just working on a single species, at the end of the day, is probably not going to be enough. I think what we're really going to need to do is move towards the direction I think that we're trying to in my lab anyway, is ask bigger questions, more complicated questions in natural systems to really get a handle on how biological communities are likely to be affected by some of these changing environmental conditions. And with that, I should acknowledge a whole lot of people. As I said, Mark Denny, Luke Miller, Lars Tomanek, and I forgot to mention my collaborator up at Humboldt State is Paul Bordeaux. He's faculty up there in biological sciences, so we're working together to do the Humboldt project. as I said, a ton of students. I think I've had 6 master students get their degrees based on projects that I talked about here, and a whole lot of undergrads went up to Hopkins for the summer. We would take, for 3 summers, we took 4 or 5 students, CSULB students, up to Hopkins, and they spent 10 weeks up there doing research. And as Julie mentioned, I've been funded by primarily the National Science Foundation for this work, and then the CSU as well. Thanks.
Well, thank you, Dr. Allen. I hope you'll entertain some questions.
Absolutely.
Why don't we, any questions from the audience?
Shout them out.
Have you had a chance to look at the mitigating proteins in the muscles up around Humboldt?
No, we don't actually have funding for that anymore. So what we will do probably, because we can basically freeze the tissue and then look at it later. And so what we will do is, as we do some of these experiments, we will take samples to then put in the freezer and hope that eventually we could do that. Yeah, basically. I mean, we'll see what happens when the people that are up at Cal Poly are finished with their analyses. I mean, if we get some really interesting results out of that, that tends to make the funding agencies more interested in doing the next project. So, yeah, we'll see.
As a way to mitigate the lower pH and higher temperatures, is it possible a technique and a business can be formed to make money to pump the upwelling up?
I don't know how you do that. AltaC probably can sort that out, right? The engineers there. So, there's somebody down here?
Yeah, I was wondering why you chose Abalone Cove. Is that because it's so clean?
Because I started working with Alan Miller when I got here, and I was like, I know where Abalone Cove is. So yeah, pretty much. But I mean, it is a really nice site.
Yeah, it's an A+ beach.
Yeah, I mean, I also, not associated with this stuff, but I will also work with a group called Marine, which stands for, I forget, Multi-Agency Rocky Intertidal Network. So it's marine.gov, actually, you can check them out. But they do monitoring up and down the coast. I think it's where like 100 plus sites. And so I added Abalone Cove as one where we do it used to be twice a year, now it's once a year. We take photos of the same quadrats over time. And so we're tracking kind of changes in mussels and algae and barnacles. And so because I was doing that, and I do the survey with Julie and Suzanne and Alan as well at Abalone Cove in a different area, it just seemed a natural place to kind of add it.
Did you get any static from California because it's a marine preserve?
I'm currently getting my collecting permit revised right now, and I don't think so. I mean, no, I don't think so.
Because I was down there culling some sea urchins,
Yeah.
and I got in trouble for it.
Yeah, they don't like that. Yeah, no, everybody's been pretty supportive. The city of Rancho Palos Verdes, I get insurance through them and stuff. So, no, I've not had, cross my fingers, but it's been pretty good.
All your research and data that you're doing on mussels, will this data be useful to the mussel farm that we just got started off?
Yeah, maybe. I mean, I think that, depends. I mean, I think in the sense that, in a broad sense, because I work in the Intertidal, and that obviously is a little bit different than a culture farm. But really at the end of the day, if it is sort of stress tolerance is related to food availability, and in other words, the muscles' ability to deal with stress, if they have access to food, maybe that's not stressful, and so forth, that would probably be relevant, I think. I don't know how much they can do about the food, but you might figure, if you're trying to figure out where to site something, you might put it in an area that has historically higher levels of phytoplankton or something like that, but I don't know.
The rock that you were drilling holes into?
Yes.
What did you do with the holes when you were done?
Well, we're not done, so the plates are still out there. I mean, we don't have funding anymore for that, but we are still sort of piddling away at it. When we are officially, because Luke, my collaborator, well, Mark Denny is still up there at Hopkins. He's actually going to be director starting in January, I think. And then Luke still works out there as well, because he's about 45 minutes away. So when we're all done, finished, then we'll basically we kept all the rock plugs. We marked them so we know which ones go to which holes, and we'll cement them back in. Yeah. So, but we don't want to do that until we're totally sure that we don't want to ever use them again. So.
Does coral bleaching have any effect for low pH, or is that just temperature?
It's both. Yeah, it's both, and it's sedimentation, and yeah, I mean, there's all of the above. But yeah, no, bleaching, I think that is actually probably the most, I mean, it's just not accessible to me. But yeah, if you want to look at the interaction of multiple stressors, that's probably the best system right now. Yeah, and it's happening quickly now. Yeah, so no, I think pH and temperature together and because the thing is, what I think we're seeing too is that those factors might then lead to you're more vulnerable to disease, for example. So disease is becoming an issue on some of the reefs as well, perhaps because of the temperature and pH issues, that if those weren't there, maybe they could fight off the disease. So yeah, it's a complex problem for sure.
So this is a Julie Passarelli question, but those limpets tend to be heavy carriers of Cyparia parasites. I think an obvious ancillary question is, what's the temperature doing to the infection of these things with parasites?
So we have a lot of frozen limpets if Julie wants to look at them. Yeah, I don't know. It's a great question, actually, and we just froze them.
That'll affect their growth.
Yeah, absolutely. It's a totally reasonable alternative hypothesis that I have no answer for you. So I'm going to say I don't think it matters, honestly. No, I'm just kidding.
Why did you pick limpets?
Because we wanted something, because this is I probably wasn't very clear about it. It's a perfect system for letting us manipulate temperature in the field. So that's actually why we got funded by the National Science Foundation, is because nobody had ever done that before. Well, mostly people didn't deal with variation anyway, but they definitely didn't do it in the field. And so because we could this is Mark's idea, basically. It was like how we could manipulate it, and then we had to come up with a project that we could piggyback on top of our manipulation, basically. And limpets were perfect because they sit there, you can mark them, and we just took pictures of them, so we didn't have to touch them, so we could leave them out there for months. And with the fences, they mostly wouldn't crawl away. And because of that big foot that they used to stick on the rock, their temperature, and Mark had shown this previously with an actual engineering model, showing that their temperature is the temperature whatever they're sitting on. And so as long as we could manipulate the plate temperature, we knew what the limpet temperature was as well. So it was just a really great system for that. But that's why we switched to mussels in the lab, because with mussels, it's really easy to manipulate. We can get cultured phytoplankton in bottles, and you just pipette out the amount you want. So it's really easy to control food, whereas we couldn't figure out how to control microalgal levels on the plates in the lab. I mean, it wasn't a good lab study system, if that makes sense. So that's sort of why we switched, is like, well, what lets us answer the question we want to answer right now? Not that we care so much about limpets per se. I mean, they're great, but. Yeah.
Did you look at reproduction rate as a life history trait?
No, we stayed as far away from that as we could. I mean, we just weren't sure even how to approach that. So, yeah, I mean, I will say that with the stuff I'm doing with Paul Bordeaux up at Humboldt, we're talking with Christy Forsgren, who is at, she actually gave a talk here sometime. Yeah, I don't know when. Yeah, I would say not that long ago, right? So she does reproductive biology, mostly of fish, but she has some side projects working with mussels. And so we're actually talking with her about, I think she's gonna assuming we don't get funded this year, we're going to probably try again and try bringing her into the project as well to deal with reproduction for the mussels. Because honestly, that's actually probably even more than growth. Reproduction is the first thing you're going to give up when you don't have enough energy. So yeah, it's a really important thing to look at, and we didn't because it's hard. But yeah.
Is there any way to know what the pH was in the ancient past, and whether some of the massive species were correlated with a rapid change in pH?
I don't know. I feel like there should be, right? I mean, I don't know. That's an interesting question. I mean, I feel like there must be signals for that. I mean, there are for so many, for oxygen and other things. So, but I don't know what those techniques would be, and I don't know that anybody's actually tried to correlate it with some of the marine extinction events or anything like that. So that's actually a really interesting question. I will say that this is something that people have really not been thinking about for that long. the pH issue. And so, yeah, that's an interesting question. I don't know.
How about one more question? We got one more question out there.
Hi.
I just wanted to know, you started mentioning about the tissue samples that you guys were taking. How did you guys go about the tissue, measuring tissue growth?
With the limpets?
Yeah.
So indirectly, actually. So what we did is we took pictures every month. We had this camera stand that we would just sit over the plate, you just take a picture, and then we could go with an image analysis program. You can draw a circle around the edge of the shell margin, and then we could get it from one and we had tags, individual color and numbered tags on every limpet, so we could basically track its growth from one month to the next. And we just had a couple of hundred limpets that we had collected over time, and so we knew if your shell is a given diameter, we basically could estimate how much tissue was underneath it. And so that we could just back calculate from that. Yeah.
Dr. Allen, thank you so much. A token of our appreciation from the Friends of Cabrium Aquarium and also the Alta Sea folks. So yeah, he's looking for the wine now.